This is Dr. Launen from Keene State College, and this is the last section of the lecture on the basics of transcription for genetics class. And what we're going to talk about now is what happens particularly in eukaryotic cells to RNA after it is produced and before it's actually ready for translation. And so the three things that we're going to look at are processing of RNA, which includes 5' capping, 3' polyadenylation, and removal of introns. So here's a simplified version of a eukaryotic gene. And we've got coding sequence that's broken up by introns and peach, so intervening sequences, and the coding sequences themselves are the exons, and they're in kind of like this lighter, winey kind of green. We have our promoter region, which is important in the initiation of transcription. We already talked about that. And so when RNA polymerase synthesizes an RNA transcript according to this information, this template information, you get a molecule that's quite long, longer than the final messenger RNA, and we refer to it as the pre-mRNA or the unprocessed transcript. As soon as the RNA is produced out of the transcription bubble, the first thing that happens to it is it will get a 5' cap, because that's the end of the molecule that's actually poking, out of the transcription bubble and therefore available for modification. And that's going to be at the 5' end, obviously, of the messenger RNA or the pre-mRNA. As this information comes spooling out of the transcription bubble, the introns will be getting spliced, which means to remove them and connect together exon regions. And then finally, the last thing that's going to come out of the transcription bubble is the 3' end, and that's going to give us the opportunity of polyadenylating that end, which is to add multiple adenines in a row, a unique feature to eukaryotic messenger RNA. And so when you do all of that modifications, you're left with a shorter messenger RNA transcript that is ready to go to the ribosome or ribosomes and be translated. And scientists first determined that this kind of processing was happening by noticing that the length of the final messenger RNA molecule was longer than the initial predictions based on the gene coding sequence. And so they figured out processing because they were trying to understand why we saw these differences in lengths in the whole gene and the final messenger RNA molecule. So let's look first at 5' capping. So here is what's supposed to be the end of an RNA strand or sequence or chain, and it's the 5' end. So that's the part that's going to come out of the transcription bubble first, because we make new RNA growing at 5' to 3'. Here we've got a cap, and this is a cap that's made out of a molecule called 7-methylguanosine. This is a methyl group, CH3, and it's been added to this guanine nucleotide, which makes it a little bit unusual and very stable. And all of that structure is connected to the 5' end of this RNA strand with a rather unique and interesting chemical bond that incorporates 3 phosphates within it. So why do cells do this? It seems like without 5' capping, messenger RNA molecules aren't that stable, and maybe more importantly, they don't bind to ribosomes effectively. And if messenger RNA can't bind to a ribosome, it can't get translated, and then the cell can't make proteins, and that can be deadly for the cell. So a big reason that we do 5' capping in eukaryotic cells is to enhance the ability of ribosomes to bind messenger RNA. Right, so sort of skipping over the middle stage of splicing straight to the last stage of mRNA processing, which is polyadenylation, many adenines. And that's going to happen at the 3' end of the messenger RNA transcript. So here's the messenger RNA transcript. Here's RNA polymerase, and it's chugging down this gene, doing RNA synthesis. This is the gene, okay? And we've already got the 5' cap on. Splicing to remove intronic messenger RNA is occurring. And then what happens is the RNA polymerase will travel through a sequence that's called the poly-A signal. And that sequence recruits in some proteins that are going to identify this sequence, the poly-A signal, encoded here, and then present in the RNA transcript here. And those proteins are going to cleave or cut the messenger RNA molecule at that position. So all of this is just kind of like wasted or recycled at this point in time. And after this is cut, that same protein complex will add adenines to the end. It will synthesize a string of adenines. It'll polyadenylate. And so why do this at all? Like, why add polyadenines? Why add multiple adenines? It seems to help in allowing the messenger RNA to get out of the nucleus. So it allows efficient export of messenger RNA from the nucleus so it can get out of there and reach the ribosomes and be translated. It seems to stabilize the messenger RNA molecule, so it protects it from the kinds of enzymes that would otherwise damage it, which are called exonucleases, enzymes that will chip in on nucleic acids from the ends. It also seems to play, the poly-A tail seems to play a role in helping to initiate translation. And it also seems to help with the termination of transcription. So this process is happening right at the end, and it seems to help end transcription in eukaryotic cells, a process we don't really actually know that much about. Right, so that's two of the three ways that messenger RNA is processed in eukaryotes. The third is maybe the coolest, and it's the process of splicing. So splicing is a process whereby we remove introns from messenger RNA and connect together exons. So here, metaphorically, what you're doing before you splice the messenger RNA, you might have like this gobbledygook in terms of genetic information that the cell can't really decipher. And after splicing, you've actually got information organized into sort of sensible coding sequence. So we go from this gobbledygook to an actual message. Here you may now tip the pot and sip the tea. In the actual genetic world, it would be make these particular, select these particular amino acids and connect them together into a polypeptide. So what does this work? These really cool cellular structures that consist of both protein and RNA. They're called SNRPs, so they have like one of the funnest names in biology. And there are a set of SNRPs, U1 to U6. Each individual one is built by combining small nuclear ribonucleoprotein particles, little proteins, and small pieces of RNA, snRNA. Okay, so little small nuclear ribonucleoproteins and small nuclear RNAs combined make up SNRPs, and a set of SNRPs will allow the cell to splice messenger RNA. So here's a picture of what that might look like. I'll orient you a little bit better there, but here's our pre-messenger RNA molecule, and we want to get rid of this intron, and here are the SNRPs. So they're made of little proteins and little RNA molecules. They will be attracted to sites at the exon-intron boundaries, these sites right here. And these are called splice sites. And they're very, very important biologically, because if we don't remove the intron, then this becomes coding sequence and we could get a whole new or different protein, which I'll talk about more in a moment. So the SNRPs will recognize these boundaries, attach to them, pull them together, cut at those locations, and then catalyze the formation of a closing or sealing or repair phosphodiester linkage to get an intact messenger RNA molecule. So a little more about what I was saying with regards to what happens if you don't splice accurately. And actually, that's a really cool thing to think about, because in eukaryotic genomes, one of the things we've discovered in the last decade or so is that there is a heck of a lot of what is called alternative splicing. So here's a pre-messenger RNA molecular transcript, okay? And all of the colored areas are exons, and the white areas are introns. So if things were simple, what would happen is all of these introns would just be removed every time this pre-messenger RNA molecule is made to get one protein product, okay, one messenger RNA transcript that then corresponds to one protein gene product. So one gene would correspond to one protein. And it turns out that's just not true at all. So in fact, in different cell types in multicellular organisms, different introns will be spliced out, which results in different messenger RNA molecules and therefore different proteins. So in this way, one gene can encode more than one protein. And here's the classic, example learned by genetic students all over the world. It's the calcitonin gene-related peptide gene. It's a bit of a mouthful. So all of this is the gene name. And it refers to the gene that encoded all of this pre-mRNA transcript. So if that gene is present on, I forget what chromosome, but it's in the human genome, which is in every single cell that we have. And in the cells that are found in our thyroids, what will happen is you will make calcitonin by splicing so that you connect together these introns, this one, this, and this. And that gives you a protein hormone that helps us regulate calcium, which is really important for heart and other muscle function. In our other cells, in our neural cells, for example, we will splice differently so that we make a protein called CGRP hormone, and that's this here. And we get that by splicing together this, and this. So we won't fool with this at all. We'll just ditch it all, treat it as introns. And that's actually really important in vasodilation. So it affects if we feel pain or not, and it apparently plays a role in migraine headaches. So we have one gene, but it actually encodes two different final messenger RNA molecules and therefore proteins, some that are used by some cells of the body and others in different cells. And this turns out to be a key to a riddle that was discovered, after 2001, when the human genome, draft genome, was first released, and in 2003, the final genome was released. And since then, we've been studying it like crazy. And what we've found is that although we know by studying the proteome, the total protein complement that human cells can make, that we are able to make at least 90,000 proteins, if not more, we only have about 25,000 genes, which shows you there's not a one-to-one relationship between gene number and protein number. And so one of the reasons that can be, one of the explanations for that is alternative splicing, such as the calcitonin gene-related peptide and calcitonin example that I just gave you. So a dark side to this topic of splicing is that it turns out that in humans, a lot of mutations that give rise to disease are mutations that affect splice sites. So we need splicing to work very precisely. And when you have a mutation that changes a nucleotide at a splice site, at an intron-exon boundary in a gene, that can result in some pretty severe alterations that can be very harmful to the human that's experiencing that. So with that, this concludes the overview lecture on transcription for this class period. Please be sure that you're doing the associated worksheet and watching all associated supplemental videos that have been chosen for you for this topic.